So, welcome to my channel Brushup Zoology, and today we would be learning about cloning vectors as biotechnological tools. What are cloning vectors? Cloning vector, it is a small piece of DNA which is derived from a virus, or it could be derived from plasmid, or even from a cell of higher organisms like eukaryotes. For example, yeast. And vector is used to transfer a foreign DNA, which is called as insert into the host cell. And this is a technique which is the cloning vector. It is useful in, you can see, say, DNA recombinant technology. Okay. So we have two things in main two things in recombinant technology. One is we have a insert, which is the DNA that is called as which is the gene of our interest, whereas it has to be fixed or it has to be joined with the vector. Vector is a kind of vehicle which would carry and then it would multiply. It would be introduced into the host cell and that would multiply to form the clone. And such a DNA which is formed by DNA recombinant technology, it is called as chimeric DNA. Right? So, what are the characteristics of a good cloning vector? There are certain characteristics a cloning vector must have. It should have these features. First is ORI. What is ORI? ORI means origin of replication. That means whatever DNA which has to be taken as vector, it should have a site which is called as origin of replication, where the replication would be initiated. Such a site where once the foreign DNA or the insert it is joined and it can initiate the replication of that segment. So that is called as ORI or origin of replication. Then we have another features which a which a cloning vector must have, that is the cloning site, also called as the recognition site. So ORI, as I said, ORI, when the insert it is integrated to ORI, it would replicate along with that of the, let's say, the vector or the vehicle DNA within the host cell where you have introduced it. And the sequence also controls the copy number of the linked DNA. So a vector, so usually a vector has high copy number per host cell. Second, I said, is the cloning site. It is also called as recognition site. It is that site, it is that DNA segment within the vector where the DNA insert can easily be attached and it can be easily removed. For example, let's say here I am showing you that suppose this is the vector and it has a site which I said it is ORI, that is origin of replication, where the origin would of which where the this is the site where the replication is initiated at one point, whereas it would have another site from where it would have certain sites from where this insert DNA it could be easily removed as well as it could be added with the help of certain enzymes which you know which are called as restriction endonucleases, right? Enzymes. So therefore that is the name recognition site, it comes from the restriction endonucleases, means that that particular site it could be recognized by the endonucleases and they can cleave it, and this particular using these enzymes, particular specific restriction endonucleases, these inserts could be included, added, or they could be removed at those sites. Furthermore, a vector must wear at least one restriction site. It should have at least one restriction site, or let's say the recognition site. But it can have, a vector can have more than one recognition site. Means that means these many recognition sites, they are recognized by different type of restriction endonucleases. And such a small area over the DNA, suppose over the vehicle, it is being recognized by, say, let's say 3 or 4 number of restriction endonucleases. Such a site is called as MCS. That means it is multiple coding site, or it is also called as polylinker. Then another feature which cloning vector should have, that is called as marker genes. Selectable marker gene. Selectable marker gene, it is that site over the segment of DNA that would allow the selection of positively transformed cells. What it means when I say positively transformed cells, that means you have a, let's say you have a certain desired segment of DNA which you have inserted into the, along with the, onto the vector, and then you have introduced into the host cell. Suppose this is the host cell and you have inserted this, and then you've allowed it to multiply, right? So selectable marker gene is that gene. Now this gene is called as this DNA would be called as, which is with the insert, it would be called as either chimeric or it would be called as transformed, right? It is a and we need to then choose that particular, because the desirable gene, it should be incorporated with that of the vector, and then it is allowed to be cloned. And we require that particular clone of that particular required DNA segment. For that matter, we must have certain marker genes. We must have those marker genes by which we could select that positively transformed cells from the ones which have not undergone certain that kind of transformation. So that the transformants, they can be used and they could be applied for the cloning matter. So this selective marker gene, it can be of different types, such as it could be, let's say, certain such gene, such as it would be certain such a gene which would provide, let's say, antibiotic resistance. Antibiotic resistance, right? It could be that. Now, how do we explain this? That certain that particular gene which is providing antibiotic resistance